As opposed to a surgical procedure or a week-long stay at the hospital, medical drugs, especially oral ones, can be cheap and very easy to take and administer for years. They can handle many different types of health issues and be prescribed by pretty much any doctor, and these medicines improve health and quality of life for people around the world. But designing a new drug, from finding a promising molecule to running all the tests imposed by health authorities, can cost an estimated $2.6 billion. Since 9 out of 10 drug candidates never make it to market, much of this money is wasted, as decades worth of potential new drug targets have yet to be exploited. Since the 1950s, we have witnessed a decline in pharmaceutical research and development productivity, known as Eroom's law, which is Moore's law spelled backwards. Eroom's law is the observation that the number of new drugs approved per billion US dollars spent on R&D is a decreasing trend. 50 years ago, drugs were discovered serendipitously. People knew that they worked, but they didn't know how. Now, scientists and researchers start by answering how drugs work before even searching for one. They're interested in the molecular processes associated with a specific disease. It's disease pathology and target identification, wherein proteins involved are studied and select targets identified for their therapeutic activity. Once the biological target has been identified, researchers start looking at drugs that act against the disease. The drug can block the target to prevent it from continuing its activity. Because we know the structure of target proteins thanks to experimental methods such as X-ray crystallography and nuclear magnetic resonance, we have a better sense of how a drug is going to interact with a target protein. If we understand the structure of the target and the drug, we know how the drug may bind to the target. The goal is then to stop or disrupt its normal functionality. Due to huge growth in available training data, computational drug discovery and design strategies are able to factor in structural information and chemical representation to predict the key pharmacokinetic properties of drugs and their therapeutic efficacy. We can save time, money, and resources by leveraging machine learning to tough problems like drug discovery. People say that the practice of machine learning is at least 80% data processing and cleaning and 20% algorithm, and this is not different in machine learning for biology and chemistry. Our input data comes from the Chemical database, which stores millions of bioactive molecules with drug-like properties. It contains compound bioactivity data against numerous drug targets. There are many values we can use to express the concentration necessary to induce some observation. In my project, we're specifically interested in IC50 measurement, which is the concentration at which a drug can block a biological process by 50%, and particularly PIC50, the negative logarithm of its value in molar. In my project, the compounds of interest target acetylcholinesterase, a protein primarily found in muscles and nerves to break down types of neurotransmitters. Drugs that can block acetylcholinesterase are used to treat neurodegenerative diseases such as Alzheimer's and Parkinson's disease. Models capable of predicting the physicochemical and biological properties of compounds from their chemical structure are known as quantitative structure-activity relationship. Unique molecular descriptors of molecules are calculated and used as input data for our regression model. The descriptors can either be 0 or 1 to represent the absence or presence of a particular molecular feature. Our target feature Y determines the activity of a compound, where value 1 corresponds to activity and 0 to inactivity. Our 2D drawing is then represented by data in the table, and we're able to build a regression model for the predicted PIC50 values versus the experimental PIC50 values. Our model can learn the relationship between the chemical structure and biological activity to then predict the bioactivity of any drug based on its molecular descriptors. This is also useful to scientists and researchers because it can reveal what features about molecules matter the most when determining bioactivity, and which will help them design future molecules with more robust properties. AI models can both intelligently screen and design therapeutics. The transformation of drug discovery backed by machine learning may be enough to break Eroom's law. We have a tool that can quite literally predict biological activity, offloading the creative burden and covering our blind spots. In the immediate future, this technology will be used to democratize our research for molecular therapeutics in applications that were previously technically challenging and economically infeasible.